Hi everybody, my name is Douglas Shoei. Today I will be talking about eutrophication and dead zones. Every year, cultural eutrophication has devastating effects on watershed areas, causing billions of dollars in damages. Beach closures, outbreaks of food poisoning, loss of fish and shellfish drinking water treatment facility upgrades, and animal illnesses all have lasting effects and continue to worsen without proper policies to correct for cultural eutrophication. This presentation will address what eutrophication is, what causes it, what the effects are, and how it is being studied, and some methods for its prevention. What is eutrophication? Eutrophication is a process by which a body of water becomes enriched in dissolved nutrients that stimulate the growth of aquatic plant life, usually resulting in the depletion of dissolved oxygen. It affects both salt and freshwater habitats. In freshwater ecosystems, it is almost always caused by excess phosphorus. In coastal waters, the main contributing nutrient is more likely to be nitrogen or nitrogen and phosphorus together, depending on the location and other factors. There are two types of eutrophication: naturally occurring eutrophication and anthropogenic or cultural eutrophication. The first picture, naturally occurring eutrophication, is a very slow process in which nutrients, especially phosphorus compounds and organic matter, accumulate in water bodies. These nutrients derive from the degradation and solution of minerals in rocks, lichens, mosses, and fungi. The second type, cultural eutrophication, which is causing all of the damage, is often a much more rapid process in which nutrients are added to a water body from a wide variety of polluting inputs, including untreated or partially treated sewage, industrial wastewater, and fertilizer from farming practices. Causes of cultural eutrophication. Eutrophic events have increased because of a rapid rise in intensive agricultural practices, industrial activities, and population growth. Human activities have emitted nearly twice as much nitrogen and 3 times as much phosphorus as natural emissions. In developed countries such as the United States and nations in the European Union, heavy use of animal manure and commercial fertilizers in agriculture are the main contributors to eutrophication. Fossil fuels and fertilizers release nitrogen into the atmosphere. This atmospheric nitrogen is then redeposited on land and water through the water cycle by means of rain and snow. Now that we know what eutrophication is, what are the effects? Eutrophication can lead to harmful algal bloom, often referred to as HABs or toxic algae. Hypoxia is another result, or the depletion of oxygen and water eventually, if not treated, causing dead zones. Fish and other water organisms that require oxygen to breathe are suffocated, die, and float to the water surface. In recent years, cultural eutrophication has been invoked as a major cause of seagrass disappearance worldwide. A loss of coral reef as well and surrounding habitats is another major effect of eutrophication. Currently, coral reef ecosystems are threatened worldwide by multiple stressors such as changes in water temperature, ocean acidification, increases in salinity in the tropics and subtropics, nutrient loading, and hypoxia. As mentioned earlier, a major effect of eutrophication is what are called dead zones. Dead zones are low oxygen or hypoxic ocean and lakes. Because most organisms need oxygen to live, few organisms can survive hypoxic conditions. That is why these areas are called dead zones. How are dead zones formed? The diagram shown above compares a healthy water body with a dead zone. As you can see in the top diagram, there is an overabundance of nutrient inputs, also known as nutrient loading, which causes algae to grow at the surface. The algae at the surface traps the bottom body of water from getting enough sunlight needed for photosynthesis. The algae on the top die off eventually and settles to the bottom. The decomposition process of the algae requires oxygen, which it gets from the oxygen present in the water molecules. The fish and other organisms that also require this oxygen eventually die off, causing a dead zone. Another effect of eutrophication are what are called harmful algal blooms, or HABs for short. Harmful algal blooms are the rapid growth of algae or cyanobacteria that cause harm to people, animals, and the local ecosystems. Skin contact exposure from swimming or other water activities can cause irritation of skin, eyes, nose, throat, and lungs. Drinking or consuming fish or shellfish tainted with this, or even breathing in the toxins, can cause stomach pains, headaches, neurological symptoms, vomiting, diarrhea, and even liver damage. How can we detect for eutrophication? There are a few methods. The first shown here is what is called a Secchi disc. The Secchi disc is an 8-inch disc with alternating black and white quadrants. It is lowered into the water, usually from a boat or a dock, until it can no longer be seen by the observer. The depth of the disappearance is what is known as the Secchi depth, which is a measure of transparency of the water. Transparency can be affected by the color of the water, algae, and suspended sediments. Transparency decreases as color, suspended sediments, or algal blooms abundance increases. If transparency is measured through the season, from year to year, trends in transparency may be observed and monitored. Transparency can serve as an early warning of activities on the land, how activities on the land are affecting the water conditions. A second method to study eutrophication is by the use of remote imaging or satellite imagery. The most useful information for monitoring of eutrophication is water color. Chlorophyll pigment in algae is green, contrasted with the blue of the surrounding water. Dedicated sensors like the MERIS, Enviasat's medium resolution imaging spectrometer, and the MODIS moderate resolution imaging spectrodiameter can indicate areas which are seeing surface algae growth. A third method for studying eutrophication and early detection of algal blooms is the hyperspectral imaging sensors. These sensors can be put on several aerial platforms such as fixed wing, rotor wing, drones, airplanes, helicopters, and even low Earth orbiting satellites. Hyperspectral imaging, HSI, is a technique that analyzes a wide spectrum of light instead of just assigning primary colors red, green, and blue to each pixel. The light striking each pixel is broken down into many different spectral bands in order to provide more information on what is imaged. Early detection of microscopic levels of chlorophyll allows scientists to better prepare the public of potential devastating harmful algal blooms. Along with detecting and monitoring eutrophication, there are several methods for prevention. Methods for prevention include minimizing point source pollution. U.S. Environmental Protection Agency defines point source pollution as any single identifiable source of pollution from which pollutants are discharged, such as a pipe, ditch, ships, or factory smokestacks. Minimizing pollution from agriculture and other non-point pollution sources is another method for prevention. Restoring riparian zones. Riparian zones are strips of land adjacent to water that are covered in foliage that act as a buffer between the water and excess nutrients. Bubblers or aerators may also be a solution. Much like a fish tank, bubblers provide oxygen into the water while stirring up the algae, which will not be able to form. Finally, I'd like to share with you some nutrient loading policies. To combat overwhelming natural habitats with excess nitrogen and phosphorus, or nutrient pollutants, the EPA is working diligently through a series of activities and environmental policies. The Mississippi River Gulf of Mexico Hypoxia Task Force, consisting of 5 federal agencies, 12 states, and the National Tribal Water Council, was established to reduce the size, severity, and duration of hypoxic zones and their effects. The EPA has collaborated with the United States Department of Natural Resources and state water quality agencies to reduce non-point sources of nutrients, sediment, and pathogens related to agriculture in small, high-priority watersheds through the National Water Quality Initiative. EPA is working with national organizations such as the Source Water Collaborative and the Animal Discussion Group to reduce impacts of nutrient pollution. The EPA is also involved in overseeing regulatory programs that contain numeric nutrient criteria under the Clean Water Act. Finally, I would like to share with you some additional resources covering eutrophication. The USGS has conducted numerous scientific projects relating to hypoxia, nutrient uploading, algal blooms from all over the United States, river valleys, lakes, and estuaries. It's a good resource to check out if you're interested. Beachcombers, which stands for Coastal Ocean Mammal Bird Education Research Surveys, is a citizen science project with an objective to obtain information on rates of stranding for all types of species and animals in Monterey Bay, California. The long-term objectives of the program are to provide a baseline of information on the average presence of beach-cast marine organisms to assist the sanctuary in the early detection of mortality events triggered by natural and anthropogenic environmental agitation, such as harmful algal blooms and oil spills. And finally, the EPA also has a wealth of information, including research papers, scientific data findings from around the country. Thanks for taking the time to listen to my presentation. I look forward to any discussions you may have in the next several weeks.